So biology is the scientific study of life. We recognize life by what living things actually do and how they survive or how they adapt in their environments and how they interact with each other and pretty much interact with this planet that we live on. And biology is a subject of an enormous scope because it's not just, you know, talking about humans, it's talking about all living entities on this planet. So there are five unifying themes that we find in biology. One is organization. This could be how organisms are organized from the molecular level all the way up to the tissues or organ level, organ system level. Information. How is information exchanged? This could be genetic information. How is that exchanged from one cell to another cell? Energy and matter. How do we consume energy? How do we get energy? Who produces energy? There's many aspects to look at there. Interactions. We are not alone on this planet. We interact with a lot of other living species, and we have to be able to live in a coexist together. And then evolution. Evolution is organisms evolving due to ever-changing environments. And as we go through this course, we will sort of hit on these five main themes. So when we look at life, it can be studied at different levels, from molecules, which is basic, uh, elements and uh, molecules like water, or oxygen, carbon dioxide, things like that, to an entire living planet. This enormous range can be divided into different levels of biological organization. Reductionism is an approach that reduces complex systems to simple components that are manageable to study. What we end up with is something called emergent properties. This is the result from an arrangement and interaction of parts as complexity increases. Emergent properties characterize non-biological entities as well. So, for example, a functional bicycle emerges only when all the necessary parts connect in the correct way. The reductionist approach studies the isolated components of the living system. To explore emergent properties, biologists complement reductionism with systems biology, the analysis of interactions among the parts of a biological system. Systems biology can be used to study life at all levels. So structure and function is very important in biology. And we oftentimes will use the saying that the structure of a particular component is really dependent on its function. At each level of the biological hierarchy, we find a correlation between structure and function. Analyzing a biological structure gives us clues about what it does and how it works. Conversely, knowing the function of something provides insight into the structure and the organization. The cell, which is a structure that we're going to spend a lot of time talking about in this course, is the smallest living unit of organization that can perform all activities which are required for life. The cell theory states that all living organisms are made from cells. Every cell is enclosed by a membrane, which is referred to as the phospholipid bilayer or the plasma membrane, and this membrane regulates the passage of materials between the cell and its environment. We call that selectively permeable, meaning it allows or selects what it will allow into the cell or out of the cell. The cells of bacteria and archaea are prokaryotic, while other forms of life are composed of eukaryotic cells. And then we have non-living entities such as viruses, which we'll talk about later on in the course as well. So a eukaryotic cell has a membrane-enclosed organelles, including a nucleus in which the DNA is packaged. Compared to prokaryotic cells, which are simpler and usually smaller, they do not contain a nucleus and they do not contain membrane-enclosed organelles. Both cell types do contain plasma membranes or phospholipid bilayers, Both cell types do contain DNA and RNA, and both cell types are able to produce and consume energies. The big difference is with eukaryotes, we have a nucleus and we have organelles. In prokaryotes, those do not exist. So here, if we typically look at a eukaryotic cell versus a prokaryotic cell, typically prokaryotic cells are about 10 times smaller than eukaryotic cells. And as we can see from the little picture there, the prokaryotic cell, there's no real internal structures compared to the picture here of the eukaryotic cell where we can see lots of internal structures. Those internal structures are going to be the membrane enclosed organelles, and including the nucleus, which is the core. Within cells, structures called chromosomes are going to be the area where we store our genetic information. And this genetic information can be stored in the form of DNA, which stands for deoxyribonucleic acid. Each chromosome contains one long DNA molecule with hundreds or thousands of genes. Genes are going to be the units of inheritance, and in more simple put terms, they are what we call a gene is going to be a coding sequence for a protein or protein product that the cell will end up making. These genes will encode information for the building of molecules that are synthesized within the cells. The genetic information encoded by the DNA will direct the development of the organism itself as well. So here, if we look, we have a sperm and an egg. These are both eukaryotic cells. However, they are referred to as haploid cells because they only contain one copy of human chromosomes. Typical human cell has 23 pairs of chromosomes, and that is a diploid cell, which means they contain two copies. With the egg and the sperm, they only contain one copy. So the sperm has 23 chromosomes and the egg has 23 chromosomes. And when the sperm and egg fuse together, they form a zygote, which is a newly formed cell that now has 23 pairs of chromosomes. And that zygote will then give rise to a new offspring, or human in this case. The molecular structure of DNA accounts for its ability to store information. Each DNA molecule is made up of two long chains arranged in a double helix. Each chain is made up of four kinds of chemical building blocks called nucleotides, and these nucleotides are going to be adenine, or abbreviated A, guanine, or abbreviated G, cytosine, C, and thymine, or T. And here we can see typical DNA structure. It is a double helix strand. It is also going to be what we call anti-parallel, which means one strand is going in one direction and the other strand is going in the opposite direction. What you will notice with the DNA molecules is that adenines will always pair up and bind with thymines, and guanines will always pair up and bind with cytosines. That's what we call the base pair matching. For many genes, the sequence provides the blueprint for making a protein or protein-like product. Protein encoding genes control protein production indirectly. The DNA will be transcribed into RNA, more specifically messenger RNA, through a process called transcription. The messenger RNA product will then go to a ribosome, where it will undergo a process called translation, and it will be synthesized into an amino acid sequence, which will give rise to proteins. Gene expression is the process of converting information from a gene to a cellular product. So here, if we look, we have our DNA strand, which will undergo a process called transcription, forming a messenger RNA strand. Messenger RNA will then be translated by reading codons of 3 nucleotides to represent an amino acid. which will then generate a string of amino acids, which will then fold upon itself to create a protein. An organism's genome is its entire library of genetic instructions. Genomics is the study of the whole sets of genes in one or more species. Proteomics is the study of the whole sets of proteins and their properties. The entire set of proteins expressed by a given cell, tissue, or organ is called the proteome. The genomics approach depends on high-throughput technology, which yields enormous amounts of data. Bioinformatics, which is the use of computational tools and algorithms to process large volumes of data very rapidly. And then interdisciplinary research teams to kind of take all this information and really kind of make sense of it. So we'll definitely be spending a chapter or two talking more about the transfer of genetic information within cells. But now I want to switch a little bit and talk about energy. The input of energy from the sun and the transformation of energy from one form to another makes life possible. The chemical energy generated by plants and other photosynthetic organisms, which are referred to as producers, is passed along to consumers. Consumers are the organisms that feed on other organisms or their remains. So, for example, you as a human being are a consumer. You cannot produce your own energy just by absorbing sunlight. You actually need to consume carbon-based material in order to survive. However, plants are able to take sunlight and convert that into chemical energy, which is used by the consumers. So here we have light energy comes from the sun. Plants convert sunlight into chemical energy. Organisms will use chemical energy to do work. And then it sort of will all get recycled back. And the cycle just continues. And we hope that we maintain a balance between the consumers and the producers and don't sort of have one side sort of overtaking the other side. When organisms use energy to perform work, some energy is lost to the surroundings as heat. As a result, energy flows through an ecosystem, usually entering as light and exiting as heat. Chemicals will cycle with an ecosystem where they are used and then recycled. Interactions between the components of the system ensure smooth integration of all the parts. This holds true equally well for molecules in a cell and the components of the ecosystem. Interactions between components that make up living organisms, such as organs, tissues, cells, and molecules, are crucial to their smooth operation, and many biological processes can self-regulate through a mechanism called a feedback loop. In feedback loops or regulation, the output or product of the process regulates the very process itself. The most common form of regulation in living organisms is negative feedback, in which the response reduces the initial stimulus. And most regulation is going to be negative feedback. A less common form of regulation is positive feedback, in which the product speeds up its own production. And this is more of what we refer to as like a snowball effect. So here, for example, of negative feedback, we have high blood glucose will stimulate the pancreas to secrete insulin. Insulin circulates throughout the body. Insulin binds the body cells, causing them to take up glucose, and the liver to store glucose, and this will lower the blood glucose levels. As blood glucose levels get lowered, you stop secreting insulin. Here, the outcome turned off the effect. At the ecosystem level, each organism interacts with other organisms. These interactions may be beneficial or harmful to one or both of the organisms. Organisms also interact continuously with the physical factors in their environment. and this environment is affected by the organisms living there. Each organism will interact continuously with physical factors in its environment. Humans, for example, will interact with our environment, sometimes with dire consequences. Over the past 150 years, humans have greatly increased the burning of fossil fuels and the release of carbon dioxide into the atmosphere. The resulting global warming is just one aspect of what we refer to as climate change as a result of actions taken by humans. Wind and precipitation patterns are also shifting. Extreme weather events such as storms and droughts are occurring more often. Heat waves, as we're currently experiencing, is also an example. As habitats deteriorate, plant and animal species will shift their ranges to more suitable locations. For some, there is insufficient suitable habitats, and unfortunately, that species may die out unless they are able to adapt to the new conditions. Populations of many species are shrinking in size or even disappearing as a result of these changes in their environments. So evolution accounts for the unity and the diversity of life. An understanding of evolution helps us to make sense of everything we know about life on Earth. The scientific explanation for both the unity and diversity of organisms is evolution, the concept that living organisms are modified descendants of a common ancestor. An abundance of evidence supports the occurrence of evolution. And essentially, evolution is going to be, we can trace back base pair changes in the DNA, where we have seen changes in protein products, which have led to changes in how cells maybe adapt to environments or react to certain environments. Approximately 1.8 million species have been identified and named to date on this planet. Each species is given a two-part name, the genus to which the species belongs, and a species name that is unique to that species. For example, humans are Homo sapiens is the name of our species. Estimates of the total number of species that actually exist range from 10 million to over 100 million, keeping in mind that we've only classified about 1.8. Organisms are currently divided into three domains: that of bacteria, which are prokaryotic cells, archaea, which are also prokaryotic cells, and eukarya, which is going to be all of your eukaryotes. The prokaryotes will include, again, both the domains of bacteria and archaea. The domain eukarya includes all eukaryotic organisms. And this includes four subgroups, which include plants, which are producers, and they produce their own food by photosynthesis. Fungi, which absorb nutrients, animals, which ingest their food, and protists. Protists are the most numerous and diverse of the eukaryotes. These are mostly single-celled organisms, and they are classified into several groups. Some protists are less closely related to other protists than they are to plants, animals, or fungi. And here we can see some examples of bacteria, archaea, plants, fungi, animals, and protists. A striking unity underlies the diversity of life. For example, DNA is the universal genetic language common to all organisms, whether you're talking about prokaryotic organisms or eukaryotic organisms. Unity is evident in the similar skeletons of different animals, and the history of life as documented by fossils and other evidence in the saga of a changing Earth that is billions of years old. Charles Darwin published On the Origin of Species in 1959. Darwin made two main points. One, that species showed evidence of descent with modification from common ancestors, and that natural selection is the mechanism behind descent with modification. Darwin's theory explained the duality of unity and diversity. If you've never read this book or this paper, it's actually worth a read, especially if you are sort of interested in learning a little bit more about evolution, as a lot of his theories still hold up today. Darwin observed that individuals in a population vary in their traits, of which seem to be heritable, meaning that they're passed on from one generation to the next. More offspring are produced than survive, and competition is inevitable. And species generally suit their environments, meaning species are created and adapted for in a certain way based on the environments that they live in. Darwin reasoned that individuals with traits that are best suited to their environment are more likely to survive and reproduce. Over time, more individuals in a population will have the advantageous traits. Evolution occurs as the unequal reproductive success of individuals. The natural environment selects for the propagation of beneficial traits, and Darwin called this process natural selection. Natural selection results in the adaptation of organisms to the circumstances of their way of life and their environment. For example, bat wings are an example of an adaptation. The shared anatomy of mammalian limbs reflects the inheritance of limb structure from common ancestors. Fossils and other evidence corroborate anatomical unity in support of mammalian descent with modification. Darwin proposed that natural selection could cause ancestral species to give rise to two or more descendant species. For example, the finch species in the Galapagos Islands are descended from a common ancestor. Evolutionary relationships are often illustrated with tree-like diagrams that show ancestors with their descendants. So now that we've talked a little bit about what life is, how life exists, and how life sort of interacts in environments and evolves, all topics which will be dived into in the coming chapters in a lot more detail than what's covered in chapter one here, which is just an introduction. I want to just shift a little bit now about studying nature. And the word science is derived from the Latin and it means to know. And an inquiry is a search for information and explanations of natural phenomena. Scientists will use a process of inquiry that includes making observations, forming logical hypotheses, and then testing them. And guess what? Sometimes the results might come back different than the original hypothesis, in which case you update your hypothesis and statements that were made might actually change. That is science. Um, you know, you go off the data that you currently have, and then you collect more data and you keep modifying. That is how science advances. So, biology begins with careful observation, and these observations can reveal valuable information about the natural world. Biologists also rely heavily on the published contributions of fellow scientists. One of the obligations of scientists is to actually publish their results and their work so that the broader scientific community has the ability to not only get that information, but potentially even test that information and confirm that information. They build on the foundation of existing knowledge, and identifying relevant publications is easier than in the past, thanks to index and searchable electronic databases. Recorded observations are called data, and we have two types. We have what's referred to as qualitative data, which often takes the form of recorded descriptions, and quantitative data, which are expressed as numerical measurements organized into tables and graphs. Inductive reasoning derives from generalizations from a large number of specific observations. Careful observations and data analysis, along with generalizations reached by induction, are fundamental to our understanding of nature. In science, a hypothesis is an explanation based on observations and assumptions that leads to a testable prediction. It must lead to predictions that can be tested by making additional observations, performing experiments. And an experiment is a scientific test that is carried out under controlled conditions. What we mean by controlled conditions is that you're accounting for all variables, and you also have controls in place to make sure that the tests that you're using or the recording methods that you're using are working accurately. Deductive reasoning uses general premises to make specific predictions. Initial observations may give rise to multiple hypotheses. We can never prove that a hypothesis is true, but testing it in many ways with different sorts of data can significantly increase our confidence in it. A hypothesis must be able to be tested. For example, a hypothesis that ghosts fooled with the death lamp can't be tested. Supernatural religious explanations are outside the bounds of science. In order to test science, or in order to test hypotheses, we employ something referred to as a scientific method. The scientific method is an idealized process of inquiry. However, very few scientific inquiries adhere rigidly to this approach. Backtracking may be necessary partway through the process. In other cases, observations may be too puzzling to prompt well-defined questions until further studies are complete. In a controlled experiment, an experimental group, the non-camouflaged mice in this case, is compared with a control group or the camouflaged mice. In the case study that's listed in your book here. Experimental variables are also features or quantities that vary in an experiment. The independent variable is one that is manipulated by researchers. The dependent variable is one that is predicted to be affected by the response. In context of science, a theory is a broader in scope than a hypothesis, general enough to lead to many new testable hypotheses, and supported by large body of evidence in comparison to a hypothesis. Most scientists work in teams, which often include graduate and undergraduate students. Good communication is important in order to share the results through seminars, publications, and websites. And researchers are publishing their papers, but not until they have been vetted by colleagues in what they refer to as a peer review process. Scientists are charged with checking each other's claims by performing similar experiments. If experimental results are not repeatable, the original claim will have to be revised. It is not unusual for different scientists to work on the same research question and confirm each other's work. Scientists will cooperate by sharing data about model organisms. So, for example, one of the models that we'll talk about throughout this course is called Drosophila melanogaster, which is the fruit fly. And this was used as a huge genetic model when we were really starting to study a lot about genetics. There are several other popular models of organisms as well, and biologists approach interesting questions from different angles. The goal of science is to understand a natural phenomena. The goal of technology is to apply scientific knowledge for some specific purpose. Science and technology are interdependent of each other. The combination of science and technology can have dramatic effects on society. For example, the discovery of DNA by James Watson and Francis Crick and Rosalind Franklin allowed for the advances of DNA technology, such as testing for hereditary diseases, something you may have done in your own personal life. Debates on technology center more on should we do it rather than can we do it. Ethical issues that arise from new technology can have much to do with politics, economics, and cultural values, as with science and technology itself. Many important inventions have occurred where a mix of different cultures ignited new ideas. Science benefits from diversity of backgrounds and viewpoints among its practitioners. The more voices heard, the more robust, valuable, and productive the scientific interchange.